All right. Hey, welcome everybody. This is Oliver Medvedik broadcasting live or fairly live with a slight quantum lag here from City of New York at the Cooper Union for the Advancement of Science and Art here at the Kambar Center for Biomedical Engineering. And I guess I'm brought to you by Life Extension Advocacy Foundation, of which I'm a member, board member, co-founder, vice president, etc. Joining me are Steve Hill and Fatima Hill. For some reason, that name just trips over my tongue. I'll have to pronounce it a few more times. And we are bringing you yet another paper in our, well, how long has our journal club been going, Steve? When are we hitting an anniversary?
I'd have to actually look back, but it's quite a lot now. It's quite a lot of episodes.
Okay.
I'll just check that for you.
Yeah, let's keep an eye out on some anniversaries. I don't know, 20th article or second year or whatever. Anyway, the paper that we're bringing you kind of pulls together a couple of interesting threads technologically and also the biology of aging as well. I'm going to share the paper right here. It is. So it's a Nature Medicine paper from the Belmonte lab, Bayret et al., and it involves everybody's favorite gene editing tool and technology, CRISPR-Cas9. So as you can see from the title, it's single-dose CRISPR-Cas9 therapy extends lifespan of mice with Hutchinson-Gilford progeria syndrome. So progeroid syndromes, are basically diseases that are quote-unquote exhibit an accelerated aging phenotype. So not everything that is found in aging is recapitulated, but a lot of things are. So, cardiovascular problems, I guess I would say maybe depletion of stem cells. I'm not sure if anybody's looked at that, but basically a lot of morphological changes that are seen with aging are seen with people with progeroid illnesses to varying degrees. There's Werner syndrome, which basically affects a helicase. Hutchinson-Gilford is, I think, the rarest and really the worst when it comes to an accelerated phenotype. Most people die in their mid to early teens, and it's very, very rare. I want to say 1 in a million. It's probably even rarer than that. And it's pretty devastating. And in this paper here, they use a therapy devised from CRISPR-Cas9. And what I like about this paper is that, unlike a lot of other papers where so this is a mouse model, by the way. So it's a mouse model of Hutchinson-Gilford progeria. And they actually, try to and succeed to a considerable extent in reversing the phenotype of Hutchinson-Gilford mice with this CRISPR-Cas9 therapy. And what I like about this is, unlike a lot of approaches where it's very proof of concept, where you have a transgenic mouse and then you basically, you reverse the mutation, and then you have a litter of pups that have the mutation reversed, and all of a sudden they're quote unquote cured. Well, sure, that's going to happen when you reverse the mutation. But the question is, how can you apply this then as a therapeutic intervention, right? You can't really do that with stop sharing. Can't really do that if you have somebody that's already born, right? You can't isolate their stem cells and make induced pluripotent stem cells and then inject them into a blastocyst after genetically modifying them and wait for the next generation to be cured. You want to cure that individual right there. You want to fix their somatic cells. And that's kind of what we're all about also when we want to cure aging, right? Or reverse it. You want to fix your own somatic cells, right? The heck with the next generation. No, we care about the next generation, but we also want to, fix yourselves. So this approach here, even though they use transgenic mice, this approach is very applicable as a therapeutic intervention because they applied this to mice that were newly born, I think a few days old. And let's see what they do here. So let's go back to sharing our screen. Okay, so I'm going to scroll down to the first figure. So, it's a Nature Medicine paper, so there's a lot of stuff crammed into here. They have a lot of extended data, which we won't really go into. Maybe we'll comment on some of it, but just a little background on Hutchinson-Gilford progeria and what causes it. So quite a while ago now, I think sometime in the early 2000s, I don't have the paper offhand, it was discovered that it's a laminopathy, which is basically a protein that is involved in, in maintaining the membrane structure. The nuclear membrane is defective. There's a mutation, and it's the lamin A protein, or the lamin A gene, which actually encodes a number of splice variants. I think the two key ones are lamin A, which is a full-length variant, and here we have in this figure, you can see these purple boxes are exons, 1, 2, 3, 4, 5, 6, 7, 8, 12. And this can be spliced in a couple of different ways. So remember from your, I guess, advanced high school biology or undergraduate biology, right? You have introns and exons. So that's in eukaryotic systems. That's kind of typical. So you can get different versions of a messenger RNA transcript in different protein sizes, right? Depending on how these things are shuffled around. And lamin C is the short version. And interestingly, if you delete lamin A, I think the mice actually, there's a paper cited that they actually live longer than wild type. So lamin A can obtain a mutation, a silent mutation, meaning that it doesn't change the protein because it changes basically just one amino acid to another. So you have, so at this position here, which is a C, I think a C changed to a T or the other way around, you get a glycine converted to a glycine, right? Because you have codons are in triplets and some mutations, some change in one nucleic acid in one position, usually third position doesn't have an effect. So anyway, pull up your codon table, take a look. But basically what this mutation does is it induces a cryptic splice site. So these SA, splice acceptor, splice donor, so these are regions where these exons are spliced together. And this region here, it basically, by having this mutation, it kind of makes a screwed up variant of lamin A that's called progerin. And what the consequence of this is that this can't get properly cleaved. And for some reason, I don't remember why, but progerin and lamin A end up getting a little tiny lipid attached called farnesyl. So there's a farnesylation that happens. Probably it's required to target it to a specific location, but then it gets removed. And these progerins can't get this farnesyl removed, and so they accumulate, and this defective protein actually causes the nuclear membrane to be misfolded or not very, I guess, properly folded. And that, most likely affects how genes are regulated within the nucleus, and lots of problematic things happen, and the main thing is the disease Hutchinson-Gilford progeria. So they have mice here where they induce this mutation, and so you have basically, you can have transgenic mice that have this laminopathy. So what are they doing in this paper? So that's for progeria, and that's for Hutchinson-Gilford progeria. So how are they fixing this? Well, they're fixing it by basically getting rid of lamin A, and they don't get rid of this entire lamin gene, which I believe would be, I don't know if it's a lethal mutation, but it probably would not be good. But they note that, mice can live fine and live longer if they have the shorter truncated variant of the lamin gene, lamin C. So what they do is they use our CRISPR-Cas9 system, not our CRISPR-Cas9 system, but our collective scientific CRISPR-Cas9 system that's been developed over the years. And just a quick summary of CRISPR and Cas9, you've got really two main components to this gene editing tool. One are these guide RNAs. So this is denoted by these red arrows. So one is called here GLMNA2, GLMNA1. And they basically target the Cas9 or the endonuclease, which is not shown here, but they basically tell the endonuclease, it's a ribonucleoprotein complex, where to go and where to cut, right? So basically, if you express these two guide RNAs and you express the Cas9 endonuclease, you'll get a cut here and you'll get a cut here. So basically, you're going to remove this chunk, this region. So that's why they're using these two guide RNAs, is to basically have a double-stranded break happen here, double-stranded break here, and what ends up happening is, you have this region removed. these two exons can't really properly fuse, and all you get expressed is the lamin C variant. That's in theory. In practice, you might want to delve a little bit deeper into the literature of, and I think we covered a CRISPR paper in the past where, things can happen with CRISPR where they've noted you can get upstream deletions and whatnot. So anyway, you could read about if this is exactly what happens. But bottom line is that they managed to succeed in getting rid of this region and having predominantly lamin C expressed. And how do they do this is the application, which I think is pretty neat. They end up using, again, like I said, transgenic mice. So these are, transgenic. So they're kind of two crosses of mice. So one is a transgenic Hutchinson-Gilford mouse, and the other is it's crossed with mice that are expressing Cas9. Cas9 is doesn't work unless you have the guide RNA present, right? So these mice already have the Cas9 in it. If you were going to actually apply this as a therapy, obviously you couldn't have a transgenic human with Cas9 in it. You would have to deliver a vector with Cas9 and the guide RNA. I'm not sure why they did that, probably because of size. So the vector they use for delivery is Adenovirus, and it's a pretty small genome, and it's very widely used. It's a single-stranded, I believe, DNA virus. It doesn't typically, I think there's a site of integration in chromosome 19, but it can also exist as an episome. So again, you could deep dive into that a little bit more. But it's used in a lot of gene therapy trials. So it's an acceptable vector. So they managed to scrunch in the guide RNAs into this vector, and the mouse is already expressing Cas9. So this is a born pup, right? So this is already not an adult, but it's got somatic tissues. And here's here's what they're putting in there. They have a lot of stuff already in there. So the GLMNA, which is the lamin, a guide RNA, a bunch of other stuff. These are sites for integration for integration polyase site. M-cherry is basically a reporter gene, a promoter for it. So basically to see where this is going to go and if it's going to get expressed, right? So so that's the system that they have. And this figure here just shows them applying this system, and they inject it, I believe, into a facial vein. They say this is mouse, very newly born, day 2, with this laminopathy, and here is the AAV9. So this is the adenovirus with GLMNA. PBS is just a control, phosphate buffered saline. I guess this is a light image of a mouse, and then you could see a lot of glow of M-cherry getting where the AAV9 vector is kind of floating around. We'll notice later that for some reason, I'm not an expert on adenovirus vectors, but you get a lot of expression in the liver, and you get less expression in a lot of other organs, and that's going to be a consequence later when these mice later die of kind of an unrelated, maybe a related illness. But nonetheless, the mice do live longer, even if not all of the organs are targeted. So it's good to see that this therapy is working. It doesn't have to be all or none. Even though the intestines don't get a lot of expression, still. You get a significant increase in lifespan with even, partial tissue targeting. So let's go back to share screen. Okay, so talking about the liver, so here they look at different organs. So you could see the expression, you could see where the adenovirus goes. So you can see, the liver is nice and red, and, kidney a little bit, intestine a little bit, heart a little bit, stomach not so much. So spleen, so really a lot of it's going to the liver. And what they're doing here is, D is, I gotta remind myself here, efficiency. Okay, so now they do the targeting. So now they're looking at, okay, so it's the adenovirus is getting in, it's mainly going to these locations, but is the lamin A gene actually being cut and targeted, right? So here they do a PCR. So here are the 2 flanking laminate cut sites. So this area is supposedly going to get truncated. So you should see less PCR product, right, in this region. And this is the targeted lamin A, and this is a control. So you can see that what we have here, targeted LMA. So I think this is going to be the smaller piece. So they don't have a size here, so I don't really, I'm assuming this is going to be a small fragment that they're amplifying, but it's not very, yeah. Somewhere they should mention the size of that, but yeah, anyway. So you're going to get a much smaller amplified fragment, and it seems like most of it again corresponds to liver. That's where most of the adenovirus is going. That's where this truncation is, and heart also seems to be, you might have numbers for this, but again, very qualitative here. But this corresponds with what you could see with the adenovirus entering the site. This gel blot here is a Western blot, I think. So this is an immunoblot that looks at lamin A sizes, so actually protein expression. So this is guide RNA 1 and 2, and mock is basically no guide RNAs being injected, or probably an adenovirus that doesn't have guide RNAs. And these sizes correspond to lamin A, progerin, and lamin C. So you can see with guide RNAs added, you only have the lamin C variant. You can't really see lamin A or progerin. Doesn't really seem, at least in this gel, it doesn't seem to have any kind of dramatic effect for, unlike what we saw with, heart here, doesn't seem to correlate, although again, maybe they quantified this somewhere else. It seems most of the effect is in liver, right? So that's what they mentioned in the paper, that you get this robust targeting of adenovirus to the liver. You get a knockdown of, of lamin A and progerin as a result in the liver, and all you see is the lamin C. And that's what's recapitulated with the Western blots. Okay, so I'm going to stop sharing here for a moment. So that's kind of the setup, right? So their system is going into mice. The mice are, injected with the adenovirus, and it's getting to some organs, mostly the liver. And once it gets there, it seems to be targeting the site, and you get a decrease in progerin, or actually no progerin in lamin A, and you get just lamin C variant. So that system seems to be working pretty well. So I guess now what we need to do is go see what happens to the mice. How are they doing? So let's go back to the paper. Share screen. So here are a bunch of representative mice. They really have, it's kind of hard to, well, here we have mock wild type. So this is just your wild type mouse. This is what we have here. So this is so it's kind of hard to make these comparisons because these weeks are a little bit off, but you can compare it. So this is, again, this is here it's very qualitative. We'll look at some numbers here, but this is week 19.4 mouse with guide RNAs added, and this is homozygous. So I believe it's homozygous for, I want to say the lamin A, the Hutchinson-Gilford Progeria, but let me double check. It doesn't say that here. It said it in the text. Anyway, so just comparing mock homozygous, so this is no treatment with treatment, even though this is, I don't know why this is a little younger, but I guess there's a size difference in the mouse, right? Weight, so we'll just jump into these sizes, the weights over here. So we have numbers here for mice, n equals, for wild type 4, 12, and 13, for homozygous, and homozygous with the guide RNA 1, 2. And this is male mice on this side, and this is female mice on this side. And this is how old the mice are, 9 and 17 weeks. So you can see just by looking at these numbers that there is a trend, right? So that the overall weight of the mice, when you compare, the red here with the green, which is, treated mice with guide RNA versus untreated, you can see that there's a trend in increased weight or body mass. Not as much as wild type, right? But, you know, the numbers are there, the statistics are there, and you get a, you know, an overall increase in weight, which is a good indicator of health because these Hutchinson-Gilford Progeria mice tend to be abnormally light in weight. So really kind of the meat of the, you know, the paper is really in figure 4. Figure 2, getting ahead of myself. There is no figure 4. There's extended figure 4, which is where they actually look at lifespan. So they look at lifespan and then they do tissue morphology and then they do a bunch of other physiologic parameters for the mice. Body weight progression, right? Interestingly, so one thing that jumps out, and this is probably the result of how they made these transgenic mice. So one thing when I looked at this graph, I'm like, what's going on here? Because I'm going to just jump to figure 2b. The purple is Progeria control. So these are just Hutchinson-Gilford mice. The blue, which is over here with a couple of other, you know, which is Progeria Cas9 control. I don't know what Cas9 and MOC is. I guess that's Cas9 control with probably a vector that's been injected that doesn't have the guide RNAs. But you can see it's like, wait a minute, how come the control that just has Cas9 is living significantly longer than Progeria control? They mentioned that briefly in the paper, and so I would compare this curve to the orange curve. I wouldn't worry so much about this because they're crossing a Cas9 mouse with a progeroid mouse, and they're getting, you know, offspring that basically, you know, are kind of double transgenic. So they mentioned that, you know, just there's now a different genetic background, and I'm not sure, you know, and why that would cause an increase in lifespan, but, you know, this curve here is the proper control to the orange rather than this, which is sort of the baseline Hutchinson-Gilford mouse. Otherwise, you know, you're kind of left scratching your head to, you know, trying to wonder, well, why does just having Cas9 in the mouse that's not doing anything cause this increase? And the authors just attributed to a different genetic background when they did the cross, right? So we'll take that. We'll take that as the proper reason, and, you know, and we'll basically, you know, compare these two, because in any event, the orange line and this blue or green line or blue line with green dots these are really the closest genetic matches, these two rather than this one. So you can see between these two, there's a significant difference, right? So average lifespan goes from 20 weeks to about little over 25 weeks, and maximal gets bumped up from 26 to 32, right? So that's a pretty significant increase, especially since not all the organs are being targeted by this adenovirus vector. And they look at, and the numbers are pretty significant too. So there's, you know, N equals 32, N equals 39 for mice. The body weight progression, you know, so they lose their body weight much more slowly over time as well. That's an A. And then they do histology, tissue histology, stomach and skin. So, you know, you could see just at a glance, you don't have to be a histologist to see that these look thicker, right? And these look thinner. So this is Progeria MOC, and this is Progeria with the guide RNA put in. I'm assuming that Progeria MOC, these histology samples are from the Progeria with the Cas9 that's expressed in it. That is what I'm assuming. But anyway, this symbol here, which I guess is that a yen? But anyway, that refers to fatty tissue, so adipocytes. So you can see that there's way less adipocytes here. It's a thicker fatty layer. You know, there's a thicker layer of epithelial cells. So anyway, morphologically, the histology for, you know, the stomach, which again, you have parietal cells on the inside, which secrete a whole bunch of, you know, acids and digestive enzymes. It's much more thicker after, you know, treated with the guide RNA 1 and 2. So histology looks better. And, you know, just looking at physiologic parameters, right? So they do forelimb grip strength, which I guess a mouse is holding on to something, and you measure its force at which it's pulling on something. I'm not sure what it's pulling on, but it's pulling on something. Hind leg grip strength, same thing for the hind leg. So you could see a trend, right, that it goes up. You know, it goes up, and, you know, I think it says n equals 5 times 5. Not sure what that means. 5 in each group. But anyway, revolutions per hour. This is running wheel assay. So mice are nocturnal, so, you know, they have more activity at night. This is running wheel activity, dark, light, dark. So no treatment, mock treatment. And these are 4 mice, I guess, are representative mice. This one looks better. It looks like it's got a nice nocturnal split. These guys are pretty active, but, you know, pretty active at light too. But much more activity than no treatment or mock treatment, right? So, I'm assuming each of these little peaks means every time it's been on the wheel and for a certain length of time, or maybe, yeah, running wheel activity. So, the bottom line is that the mice are doing significantly better. There are some anomalies noted. The mice do, of course, still die prematurely. They die after 32 weeks, right? Normal lifespan of a mouse is, can't remember how many. What's the translation in weeks? About 2 years, that 50 to 100 something weeks. So still significantly less for a mouse, but way better than the mouse that's been untreated, right? So clearly they're living longer and their physiology is improved. They do die of pretty abruptly, of pretty kind of horribly. The way these mice die actually is their intestines get compacted full of feces, and they just drop dead basically because they can't defecate. So they have some pretty, you know, gory pictures showing, you know, bloated intestines of these mice. And the consequence of that is, as a result of probably the adenovirus, you know, it's not going to the intestines, it's going to the liver and maybe the heart. And probably the intestinal cells go as a result of this laminopathy. And, you know, and so you have organ failure at the intestinal level. So it's kind of interesting. I think it would be, you know, I think this paper is very interesting from 2 perspectives. One is that it does offer a viable treatment for people who have Hutchinson-Gilford progeria. You know, adenoviruses are used in human trials. You can inject them into people. If you can use a version of Cas9 that's smaller, not Cas9, but you can, you know, then be able to get around, you know, the packaging requirement. So you have to, you know, pack in 2 guide RNAs along with the Cas9, unlike they did in this paper, which is they had a transgenic mouse with Cas9. If you can get that into a suitable vector, then you can basically, you know, get it to at least to some tissues. And at least this paper shows that that would be good enough or an improvement enough. Certainly anything, you know, that gives better physiologic parameter, you know, improvement and lifespan extension is better than nothing. I think the only treatment for Hutchinson-Gilford progeria is farnesyl transferase inhibitors, which have some other side effects because it hits a lot of enzymes that, you know, the farnesylation of progerin, I think, is one of the key problems. But I don't think the farnesyl transferase inhibitors are specific enough to just target that. They target a lot of other farnesyl transferases, and that comes with its own side effects, which I don't know off the top of my head, but I don't think it's a very good therapy. And certainly this is the first other very viable therapy that I've heard of for a progeroid illness. A lot needs to be done. Should it be fast-tracked? You know, human therapy trials. I think the authors certainly think so, and I certainly think so, especially if you have a disease that gives you a lifespan of 14 years. Of course, why not? So improvements certainly need to be made with regards to targeting all the other tissues, right? Not just the liver, but it's heartening to know that that's you don't need to target everything to get an improvement. The other interesting bit is from a scientific perspective, the viruses primarily targeted the liver, didn't seem to hit the intestines. They didn't have any expression in the adult tissues in the intestines. And as a consequence, you probably are now seeing a very kind of, not only, is Hutchinson-Gilford an accelerated aging, disease, I guess we can call that, from a lot of kind of phenotypic perspectives. But by having this virus kind of accidentally only target certain tissues, we're seeing this disparity of aging between tissues, right? So you might be able to have a model here where you could see accelerated aging of one type of tissue, but not other types of tissues. And how does that correlate to the overall physiology of an organism, right? So it's been talked a lot about in the literature that that you have tissues can age at different rates depending on the burden of senescent cells and the amount of stem cells that you have. It'd be interesting to see if this could be developed as a model where you can specifically tweak the aging or target the aging rate of particular tissues. And see how they function within the context of a whole organism. I don't know. That just seemed to be as kind of like an aside. I just thought that was very interesting that the mice died, but they couldn't say that they died of aging. They died of basically intestinal failure, right? But is that due to the fact that the intestine is now rapidly aged and how does the kind of the morphology of those intestinal cells look? How does the physiology, how does the senescent cell burden, et cetera, look from these, from these Hutchinson-Gilford mice that have been treated with this guide RNA versus normal Hutchinson-Gilford mice? I guess what I'm getting down to is if an organism dies because everything is rapidly senescing, right? What is it? What's the main organ that's going to kill it, right? Usually there's one thing, right? Usually everything doesn't go simultaneously, right? That's why they put down on your postmortem when somebody dies of even advanced old age, they'll say, well, died of a heart attack or died of some organ failure, but that's usually due to, that organ going first, right? But it would be interesting to see if you can regulate the clocks of aging organs as a whole animal kind of matures by using this method while kind of keeping the other organs younger. I don't know. That's just an inchoate thought that I'm still kind of working on, but that sort of popped out at me when I read this paper. Right, so I don't know if there's another example, I guess is what I'm saying, putting it in other words, of an animal that has died and has died primarily because only a certain subset of organs and tissues have rapidly aged versus all of the organs and tissues rapidly aging, right? Could you make an animal that has, so looking in the other direction rather than extending the lifespan of an animal, can you have a system where you just have a rapidly aging brain and everything else is fantastic? Or rapidly aging kidney and everything else is aging at normal? Anyway.
It is possible because we know that some tissues do age at different speeds, or can, even in normal aging.
Yeah, and there was a conference I was at a couple of years ago where it was sort of reaching a bit, but I think it was a very interesting conference where they tried to implicate certain diseases like HIV to be, you know, I don't want to say an accelerated aging phenotype, but basically by having your population of cells depleted very rapidly because you have a high turnover of helper T cells, eventually you exhaust the population of hematopoietic cells that can you know, replenish the immune system, and then eventually you have immune system collapse. But the rest of your tissues are fine, right? So, you know, it's sort of a very kind of a hyperatrophy of you know, that you could consider, you know, once we have all the pieces and have a grand unified theory of aging, if it's due to, you know, if one major reason of aging is through a variety of insults, you have a depletion of stem cells, then you could say, well, that's probably a hyper accelerated aging phenotype.
Well, I mean, in a very real sense, diabetes is kind of that, because it's accelerated aging in certain tissues, you know.
Yeah.
So yeah, it's perfectly plausible. It's also when people smoke a lot and they find that certain tissues are more aged than others. Obviously the lungs, that gigantic crash. I hope that wasn't important.
I hope so too.
It sounded like a lot of sample jars being dropped. This was how the plague in New York began. The virus
yeah, that's
a movie.
That's not a sound you want to hear in your BSL-4 facility. What was that?
It's like this is how it began, the Great Plague.
Anyway, another aside for this paper is that simultaneously there's a different lab, I think Fernandez et al. It was mentioned in this paper, where they basically also targeted Hutchinson-Gilford progeria. I think they used another mouse model, and they used, I think they also used CRISPR-Cas9, but they used a different Cas9 that was from Staph aureus, I think, and it was a peritoneal injection rather than a mouse base vein injection. And I have not read that paper, but that has come out, I think, right about the same time as this paper came out. So two labs working neck and neck. And let me see, let me scroll through here. Where is that? Somewhere in the, they cited this paper. Oh yeah, Olaya Santiago Fernandez, Nature Medicine, also Nature Medicine, in the press. Okay, so that's another paper, same topic.
And we have a comment from Mark Millennium, which I've put in the chat on the side.
Mm-hmm.
50% of aged cardiac progenitor cells are senescent. The heart would be a good organ to target.
Yeah, well, I mean, at least it seems that the adenovirus here, at least in the mice, it seems to be not to a very high extent, but it seems like the second highest organ being targeted is the heart, cardiac cells. So, you know, again, this is only for, you know, targeting a laminopathy, targeting progerin. I'm not sure if there's anything else. Yeah, I'm not sure if other than Hutchinson-Gilford progeria or another type of illness that basically could be cured in such a way. Because number one, it's a very rare illness, but it was very amenable actually to CRISPR-Cas9 because they didn't need to actually insert anything. They just removed something, and CRISPR-Cas9 does that very well. So, so just getting rid of that variant was enough. I don't know if there's anything else off the top of my head that would be so amenable within the realm of aging or accelerated aging, you know, phenotypic illnesses that that you could use the CRISPR-Cas9 system in such a direct manner. Another question from Melvin Stutt. or Stutt, currently doing a research paper on the use of nanoparticles to cure Progeria. Do you think that in the future it will be possible to combine CRISPR-Cas9 with different kinds of nanoparticles administered? I don't know much about nanoparticles, but it's about packaging, right? So it's just a matter of how big your nanoparticle is and what your nanoparticle is gonna, I mean, there's, right? So it's basically size and delivery, right? So is your nanoparticle going to have a receptor or be able to bind where it needs to bind? And also, is it large enough to contain, you know, several kilobases or several tens of kilobases of genetic material? And is it non-toxic? If all of those three things can be checked off, then certainly, yes, it could definitely be applicable. Yeah. So the good thing about these types of gene therapies is that all of these components are very orthologous, so you can work on them independently and then put them all together into a functional package. So yeah, yes. But again, I don't know, you know, I don't know how. Certainly, this is, you know, I'm excited about this because this is a very good near-term attainable therapeutic. It has definite overlaps with aging, you know, albeit, you know, it's very focused on a mutation of accelerated aging. As far as actual human, you know, global interventions that have to do with sort of kind of regular aging, you know, using gene therapies and CRISPR-Cas9 systems, I'm trying to stretch my brain here to see how that would work, but I would think that, like, you know, I would like, if I were going to kind of put on my radical futurist hat for a moment, I would probably imagine a system where, you know, we look at millions of people's of genomes, right, and take a look at all of the mutations and SNPs and all the key ones that basically, or the key genetic differences that basically allow some people to live to a very, very robust age, you know, 100 plus, and still be able to run in marathons at 90 plus, and, you know, and have you know, very high, good homeostasis and resiliency, and try to recapitulate as many of those changes as possible throughout all of our somatic cells. And I don't know how many genetic changes and modifications, you know, without changing who we are, right? We don't want to become that person. We just want to become, inherit or adopt through horizontal gene transfer that individual's healthy traits, and use perhaps some, you know, some new super CRISPR-Cas9 system with some sort of radical delivery system to basically tinker with every cell, or at least all the somatic stem cells in our body, right? And kind of through an intervention to get them to be an improved version of ourselves, right? So I don't see that happening anytime soon, but it's certainly, I can certainly imagine how such a therapy could work, if not for the exact details. And in a very crude sense, we have something like that working here, right? In a very, 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 very crude sense, we actually have that first baby step here. We're, you know, changing lamin A or progerin into lamin C, and we're getting a significant lifespan extension in an accelerated aging phenotype. So it's, you know, it's helped these mice. It's working, and I think it certainly gives a roadmap for future interventions. Yeah, I mean, I don't think you can really ask for much more for a first in-class paper of such an application. And I'm hoping to see, you know, a lot more in this direction. Particularly in human trials, at least for accelerated aging phenotypes.
Yeah, and there is also the other consideration. If these therapies are eventually translated to humans, it does sort of pave the way, as it were, for more confidence in gene therapies. And there is a certain amount of sort of, should we say, risk aversion and caution at the moment towards these new types of therapies like gene therapy. And of course, when you add the whole idea of targeting aging, it freaks some people out. So perhaps the tools that we need in the defeat of age-related diseases will be developed for other diseases that are not necessarily age-related, or have an overlap like progeria. So I could honestly see them going, "Oh yeah, it's a rare disease. Let's fast track that," because they like those sorts of things, don't they?
Yeah.
But in the broad sense, it does then serve to say, look, there were some setbacks about a decade ago with Jesse. Can't remember the surname now. The person that sadly.
There's a number of people who got cancer. There were a number of cases that people dying from.
It was
therapy.
It was actually I think it was wasn't it a reaction to the AAV or something, and unfortunately,
there was a shock response. The body's immune system went into overdrive.
So it's a lot of people say, oh well, gene therapy isn't quite ready for prime time, but sooner or later I think it will be, and I think perhaps diseases like progeria will pave the way.
Yeah, and also there's also the backdoor gene therapy, which is modifying certain subsets of your T cells, for anti-cancer treatments, cytotoxic cells, for example, that have been trained or basically genetically modified to have new receptors and then put back into the patient. And this CAR T therapy is really progressing rapidly, considering that it's a cell-based therapeutic that's also a genetically modified cell from the person's body, and that's being adopted really, really quickly. So the statement that I made earlier about I could imagine putting on my futurist hat for the moment and pontificating about doing because again, papers have been published in the past showing that the Cas9 CRISPR system, you could do, you could multiplex sites. You can hit multiple locations at once. I don't know what the limit is. Certainly, there's some papers which looked at dozens of sites hit at once, depending on how many guide RNAs were expressed. But let's say the sky's the limit, that you can potentially hit thousands of sites, right? Depending on how you design a system. Well, you could, going back to what I mentioned about different organs and, aging at different rates, certainly the immune system is kind of the low-hanging fruit because you can isolate your hematopoietic cells, or you can isolate regular cells and cause them to become induced pluripotent stem cells and make them into hematopoietic cells. And once they're in a dish, you've got time to basically knock in, knock out, basically do as many point mutations as you can rather than inside of a patient with adenovirus. So, it's certainly plausible in the not very distant future to basically make an improved version of your immune system based on, basically genocopying somebody who's a supercentenarian and using that as kind of a template to basically improve your hematopoietic system, and then put that back into your system. And at the very least, at that point, you would have a supercentenarian immune system. Now, of course, the rest of your body is not going to be genocopied to that extent, but I don't know. That's, I could see things like that, happening.
Yeah. And other things like adding enzymes to certain immune cells that we don't normally have, allowing them to break down insoluble waste, for example, that we can't normally metabolize. That could be done ex vivo, things like that. So I think there is definitely scope for ex vivo initially. a lot of cellular partial reprogramming approaches that are we're hearing a lot about now. They're sort of starting off, quite a few of them. They're starting off ex vivo. They take your cells, they roll back the aging on them, and then they put them back. So it's more controllable. So I think you might be right. It'll probably start like that, ex vivo, and then sort of progress to in-body modification. I don't know when we're going to get our supersonic frog legs, or night vision, or UV reactive skin that allows us to photosynthesize. But I'll take cure aging. I'll take that as a first.
I think there's a body hacker site somewhere out there where people have given themselves night vision by injecting a chlorophyll-based molecule, or not injecting it, but basically dropping it into their eyes, and it gets into the retina, and it basically is good at reflecting photons. But I wouldn't try this at home, and I'm not going to post the link, but people could Google it and find it. Side effect is that it makes your whole eyeballs basically like jet black. So
yes, Vin Diesel
style. Yeah,
I've seen that film. Actually, it was a woefully underrated film. That was.
I didn't see it.
Yeah, it's really good. I mean, the second one is not bad. I think the third one was all right, but yeah, the first one was the best. It was weird.
Okay, well, I will put that on my list.
Anyway. But I got the reference anyway about the black eyes. Pitch Black is the film.
Okay, well, anyway, good film recommendation there from Steve Hill. I'm going to go check it out. So I guess that we've covered the paper. I guess that's it. I think the next paper that we're going to be doing is going to be on a reversing crosslinks paper, right? There's one paper that people are interested in hearing about. So talking about new enzymes. Not exactly sure what the approach is in this paper, but it's a way of reversing extracellular matrix crosslinks, which is a problem with with aging, because we talk about cells, but cells are part of the milieu. The other part is the non-cellular stuff that basically is the magical glue that holds everything together and tells cells where to go, and that also ends up being aberrant. And so we need to not just work on cells, but we need to work on all the in-between stuff as well. And I believe that paper that we're going to do next, and we'll let you know what it is soon enough, that is what we'll be covering later, I believe at the end of May sometime.
Yeah, it'll be around about the usual last week or two of May. And it is, it's not glucosapine, unfortunately. It was two other kinds of crosslinks, and it's from Yale, the Spiegel lab at Yale. And I think it was a result of, at least in part, funding by the SENS Research Foundation, because they've been doing pretty much the bulk of the work on AGEs. There aren't many labs that are looking at these AGEs at all, which is shocking. But yeah, pretty interesting breakthrough. And for me and a lot of people, it was, okay, it wasn't glucosapine, but what was interesting was it was proof of concept that, yes, we can find these enzymes. So I'm going to look forward to that one.
Excellent. Well, I guess that's a wrap for today. And thank everybody for their comments. And hopefully we'll get more comments next time. And it's been a pleasure reading these papers and discussing them with everybody out there.
It has indeed. And and thanks for everyone who dropped by today. And of course, thanks very much to Lifespan Heroes, our monthly patrons, who support us every month and make shows like this possible, and webinars and all the other wonderful things that we're doing. So thanks very much for your support there. And if you'd like to learn more about that, you can visit www.lifespan.com/hero, and that tells you a little bit about the sort of things that you can get involved with. And there's even the opportunity to come and join in as well. So yeah. How about that? So we'll see you end of May, and we'll talk about some advanced glycation end products. I can't wait.
Me neither.
It is more exciting than it sounds.
We all can't wait, in a sense. All right. Take care, everybody.